Welcome everyone to another edition of the Super Spreader Seminar Series. Today is April 20, 2023. We acknowledge the land the OMNI Network's head office is located on York University. Now, the university recognizes that many indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. And so York University acknowledges its presence on the traditional territory of many indigenous nations, and they seek to have a peaceable relationship and share that and care for the Great Lakes regions. Now, about this series, the Super Spreader Seminar Series encourages highly qualified personnel, mostly including postdocs and graduate students, to present their research and to promote their ideas and share science, discuss science, and establish connections among the various branches of the network. And so it is a platform to spread scientific knowledge about One Health research framework and showcase research progress within the network scientific research community. So this seminar will be beneficial for knowledge transfer among OMNI and the emerging infectious disease modeling networks and collaborating scientific community. Now, today we are really fortunate to have Dr. Suzan Farhang-Sardroodi as the speaker, and I will just talk briefly about who Dr. Suzan Farhang-Sardroodi is. Dr. Suzan Farhang-Sardroodi is a physicist with a doctorate in evolutionary graph theory and a postdoctorate in disease computational and mathematical modeling. Her interest in mathematical modeling stems from her passion for mathematics with an application to biological reactions of the organism against foreign substances. As she progressed through her career post-graduation, she got more interested in understanding the mechanism of human diseases, including cancer and infectious diseases. She is currently a senior postdoctoral researcher in the Department of Mathematics at the University of Manitoba and a member of the Society for Mathematical Biology and Cancer Research Network, Quebec. Her research interests include computational biology, cancer, immunology, pharmacology, and machine learning. During the COVID-19 pandemic, she has led some projects in collaboration with the Canadian Centre for Disease Modeling and National Research Canada in Toronto, investigating the human immune response post-infection or vaccination. And so you realize that she's really done a lot of research. So it's my singular honor to welcome Dr. Suzan Farhang-Sardroodi to lead out and then give us her talk for the day. Okay, so Dr. Farhan, you are welcome, and you can go ahead to share with us what you have for today. Thank you.
Hi, Joseph. Sorry, let me find out. Okay, can you see my screens?
Oh, sure.
Okay. Thank you, Joseph, for your very nice and kind introduction. Hello, everyone. Today, I'm so honored that you have invited me to present some of my works so far that I have done in mathematical modeling. So I would like to talk about mathematical modeling to identify optimal dosing schedules from chemotherapy to COVID-19. So let's get my laser here. Okay, in the first part of my presentation, I would like to talk about chemotherapy-induced cachexia and the model-informed dosing to preserve limb mass in cancer treatment. So cachexia actually is a debilitating condition that causes the irreversible muscle loss during cancer and also during the chemotherapy. So and it's very common in pancreatic and gastric cancers, which is and it is a deadly side effect of the chemotherapy that causes muscle atrophy, which is irreversible. So actually, in this project that was supervised by Dr. Wilkie from Ryerson University when I was doing my first postdoc. So we tried to investigate the chemotherapy-induced muscle loss in a manner that captures the nonlinear dose response of the skeletal muscle to cancer chemotherapy. And also we explored alternative dosing schedules, agent effects, and the morphine use in chemotherapy treatment. And our aim was to protect limb mass while also targeting the tumor and controlling the tumor burden, actually. So what we did, we actually started with investigating the experimental data from the literature. We found that two different schedules have been investigated by studying the 5-fluorouracil as a main agents of the chemotherapy on 6-week-old US-bred CDF1 male mice. So in the first schedule, actually, which is the daily schedule, they have studied the lean mass for different, during the time, so for different doses of the drug injections. So you can, for example, see that for the doses that are smaller than the 20 milligram, so there is no significant limb mass or muscle mass loss. So, but however, when the doses are greater than the 24 milligram, there is a drop in muscle loss, muscle mass, and then they immediately comes back to the homeostatic level. So actually, when the drug injection stops on day 27. However, if you look at the dose 35 milligram, there is 4 of 5 mice are dead, and there is a huge actually drop in the limb mass. If we compare this schedule with the 5 days on and 2 days off chemotherapy, which is less frequent, we can see here that the doses that are smaller than 42 milligram, they approximately track the control level, which is the black curve. However, if we increase the dose amount for the 50 milligram, so 3 of 5 mice, and for the 60 milligram, all mice are dead. So in comparison with the actually daily chemotherapy, which is more frequent, we can say that the highest tolerable dose for the daily chemotherapy is 29 milligram per kilogram. However, for the 5 days on, 2 days off, it is 42 milligram per kilogram. So using this experimental data, we developed our mathematical models. So our mathematical model actually has 3 parts. The first part is the pharmacokinetic. Actually, we developed a 2-compartment pharmacokinetic system for 5-fluorouracil. And in these equations that I didn't include it in this slides, we assume 2 compartments: the drug concentration in plasma and the drug concentration in the tissue. And then the transient terms between these 2 compartments. So the second part of the model, if now we have our PK model, so we would like to actually simulate the limb mass, how we can actually simulate the stem cell lineage system for muscle loss. So in this model also, at first we did it for the healthy state. For the healthy state, we assume just 2 kinds of the cells, the satellite cells or the skeletal stem cells that are getting activated after the injury. So, and the last step of the differentiated cells that we call the myofibers or muscle loss that are differentiated from the stem cells. So again, we assume the two compartment with the ODE equations, and we assume the proliferation terms, and then for the stem cells, and very simple equations that we assume for the healthy. However, now the question was that how we can simulate the chemotherapy effect on these equations, how we can see the muscle loss on the healthy states, on the healthy equations. So studying the biology, we saw that after the 5-FU administration, so there is mitochondrial disorder. So actually the chemotherapy causes the mitochondrial disorder. And again, from the biology, we know that the mitochondrial metabolism is a key regulation of the stem cell activation and differentiation. So what we did is that in these equations that we had for the healthy, we didn't add any term to reduce the muscle loss amount, but we added some terms with the 1 minus cubic term that multiplied by the proliferation of the stem cells and the differentiation cells in order to disrupt or actually decrease the proliferation of the stem cells based on the biology. So then the stem cells and the muscle compartments suffer from reduced proliferation term via a function of drug exposure. And then the term that we actually assume is 1 minus cubic term. That cubic term is the drug exposure that we assume. The third part is the tumor growth equations. So actually, we also looked at the effect of the drug on the tumor, because we aim to control the muscle loss and also at the same time to reduce the tumor growth. So then, for the tumor growth, we already have the exponential linear equations, and what we did is that we added one negative term in order just to reduce the tumor growth of the tumor growth actually, and then that term that we added in order to reduce the growth term was the mass action term within the tissue, which is the tumor, and the drug concentration. So this is 3 parts of our mathematical model that we developed. Next, we compared our equations. Actually, we fit our models with the experimental data that we had. In order, we would like to capture the nonlinear dose response for the lean mass. So as you can see here, we first fit with the daily schedule and 4 different doses. That is a very good agreement with our fit and with the experimental data. And at the same time, we looked at the stem ratio, which is the stem ratio, because in our model, as I mentioned, we assumed the 2 different compartments for the satellite cells and the muscle cells. So then we looked at the stem ratio. So if we look at here, we can see that the stem ratio, the proliferation of the stem cells is still high after the drug injection. However, because there is a muscle atrophy, so it means that the muscle fusion actually is disrupted after the chemotherapy injection. The satellite cells still are activated, they are proliferation, but the muscle fiber fusion has been disrupted. So then we can see the muscle atrophy after the drug injection during chemotherapy. So we have also looked at the average exposure of the drug. So you can see that, for example, the blue one is the highest exposure, which is because it is for the it corresponds to the highest dose amount. We also fit our model because in our mathematical model we had 2 parameters. The other parameters were coming from our first paper that it was about the cancer cachexia, but in the chemotherapy we had 2 main parameters. In the cubic term that I mentioned. So we were trying to fit with the daily and experimental data, and then taking the average and then find those parameters. So again, we fit with the experimental data, and you can see that again, we have a good agreement with the experimental data here with the 5 days on, 2 days off schedule. Again, we looked at the stem ratio. We can see that even for the less frequent schedule, there is actually interruption in the muscle fusion, and also it is the drug exposure is highest for the 60 milligram, because this is the highest amount of the dose that is injected. So after fitting the model, now we were aimed actually to look at the different schedules. So 5 days on and 2 days off is the better better than the daily chemotherapy, because as you saw that we can actually deliver the highest doses and with the less muscle loss. However, we also investigated different schedules. So the first one was the standard weekly dose. So here actually we assumed 2 different standard doses, the 24 milligram per kilogram for the daily schedule and 35 milligram per kilogram for 5 days on and 2 days off. So during the week, during the week, actually, we for daily schedule here, you can see that we have 168 total weekly dose, and for the 5 days on, 2 days off, 175. So fixing the weekly total dose between 168 and 175, we looked at the different schedules here that you can see. So then investigating these different schedules, and then actually we aim to minimize muscle loss and also control the tumor burden by keeping the weekly dose between these 2 amounts. So what we did is that, we assumed the AUC as our main criteria actually to see, to investigate the limb mass and also the tumor burden. So during the different schedules in the standard weekly dose actually regime, so we can see the 24 daily is the best treatment, because it keeps the highest levels of the muscle mass. However, it is a little bit decreased. The muscle mass is decreased during the other schedules. And at the same time, if we look at the tumor volume, we can see the last schedule is which is the 58. 58 dose for 3 days on and 4 days off, which is less frequent, is the best treatment for the tumor burden. And at the same time, if we compare with how much muscle loss has happened here, it's not too bad in comparison with the 24 daily. So we also, in order to compare, the limb mass with the tumor control, so we considered another criteria, which is called the total therapeutic efficacy. The total therapeutic efficacy, which is actually the AUC of the limb mass over daily, and then it's divided by actually the tumor volume treated. So then we can see that therapeutic efficacy is highest for the 58, 30 days on and 4 days off. And interestingly, it is about the same for these 2 different schedules, 35 milligram per kilogram and 42 with different actually schedules. So then we decided that this schedule is the best one that we can assume with to control the tumor. Another schedule that we investigated was the metronomic schedule. The metronomic schedule actually is the delivering multiple doses per day to maintain more consistent level of the drug. So actually in the metronomic, if you can see here, it is like first we looked at the 2 different standard doses, 24 and 35. And then you can see the others are, for example, the 12 milligram 2 times a day. 8 milligrams 3 times a day in order to have the totally 24 milligram. So if you can see here, so in these bars, we can see there is a less improvement of the muscle mass because they are a little bit higher of the 24 standard dose. But at the same time, if we compare with the tumor volume, we can see that the tumor volume is highest in these schedules actually, so for the metronomic. So we can decide that metronomic also has a less improvement in actually, it has a little, better improvement actually in the limb mass, but at the same time, it has it can control, it has a less control of the tumor volume for the metronomic. So it's not a good schedule to investigate. So at the same time, if we look at the therapeutic efficacy, so it is highest for the 35, 5 days on and 2 days off, and it's not good for the metronomic schedules. These are our 2 schedules that we studied, and also we studied other schedules in our paper that I didn't include them here. So the other topic was that cachexia in the aged mice. Actually, we looked at the 2 different age mice. The first one that we investigated, and also it was the data that was for 6 weeks old CDF1 male mice, but we also looked at the 2 years old by assuming different proliferation and proliferation probability and rate for the stem cells, and also different initial values for the 2 years old. So if we look at here, we can see that, so the green and the purple bars, actually green and purple are for the younger, and the orange and the yellow is for the 2 years old. So we can see that there is a less muscle loss for the older mice, a little bit. It's not more clear, but it is less muscle loss, because we can see if we compare these bars, it is higher for the orange and the yellow. But however, we couldn't see any difference in the tumor control for younger and older. So then again, we looked at the therapeutic efficacy. We can see that the better therapeutic efficacy for the 168, which is the highest amount of dose that is delivered during a week, just once a week. So this is the results of the, actually we can say that it is for the age mice. So we couldn't see any difference for the tumor volume, but a little bit improvement in the muscle mass. And next, we also looked at the injection of the morphine concomitant with chemotherapy treatment. So what we did is that, so adding the morphine as a painkiller during the chemotherapy, actually it has a huge, we can see the huge drop in limb mass, because the dashed line here is the 5-FU plus morphine. And we can see that there is it is a significant limb mass loss by using the morphine. But at the same time, if we compare with the tumor control, there is a great tumor control, because the dashed line here is for the morphine plus 5-FU, and we can see the tumor volume has been very well controlled by that. However, the side effect is very deadly, because it is a very huge drop. And again, if we look at the therapeutic efficacy, we can see it is highest for the 168. But when we are using the morphine, so it should be very careful, because it causes the deadly effect on the muscle mass. So in a summary, our work actually highlighted the best treatment responses are those that deliver the most drugs in the shortest amount of time. It means less frequent, following the maximum tolerated dose. No significant age-related differences are captured by the AUC measurements. And inadvertent interference between pain management drugs and chemotherapy regimes may exacerbate both on-target and off-target effect of the drug. And actually, we defined an optimal schedule consistent of, actually the optimal schedules it should be considered a weighted balance between improved tumor control and also preserving limb mass. I guess it's better to take the question before going to the second part of my talk. I can see something. Joseph, should I answer or just the chat part?
Yeah, it's a question. So probably, like, if you, I don't know when you want to react to it, but if you want to finish and then we can go to the question, or you want to answer them right away. Any of them is fine, but
Okay, I'll do that after finishing. Okay, I'll answer your question.
okay. So
After.
the question is, so you want to finish before you answer?
Yeah, I guess for the first part, better to answer that, and then I'm gonna go because the second part is about the adenovirus vaccine during the COVID. Yeah, I guess better to answer. Yeah.
Okay. So I can read the question out loud. So the question is, from this mice experiment, can we say anything for humans?
So actually, our experimental data is for, yes, it is for the mice model, but we couldn't find the data for the human, but actually, so actually these schedules that we investigated, which is on mice, but also from that literature, I remember that says that it is also tested on the human. I think in Japan, they are using the 5 days on and 2 days off the chemotherapy schedule, because it has also effect on the human muscle mass. So yeah, they are, that is investigated. Also now it is delivering, it's applying on the human. Yes.
Right. Okay. So there's another question in the chat from Benas. And the question is, did you conclude morphine causes muscle loss, or is there any association?
Did you conclude morphine causes muscle, or this is no, we concluded that the morphine concomitant with the 5-FU injection causes muscle loss.
Okay.
Our model actually is predicting that. If I come back to the slide, maybe here, yeah, we can see that our model actually it causes, yes. Maybe I can come back to this.
Okay, right,
This is the model, this is the model prediction that we can see that there is a muscle loss, yes.
Okay, we don't have any other question there in the chat, so I guess you could go ahead and.
Yeah. Okay, sure. So in the second part of my talk, actually, I would like to talk about the analysis of the host immunological response of adenovirus-based COVID-19 vaccines. So in the first part, we talked about the different actually schedules of the drug, chemotherapy drug injections. And here we are looking at the different strategies in order for the actually vaccine implement. So during the early days of the COVID-19 pandemic, so there was a shortage of the vaccine. So different strategies actually were investigated in order, to overcome those shortage and then to deliver, the vaccine for the more people. So in this project that actually, this was also done during the early days of the pandemic when I was at York and under the supervision of Professor Fernan. So actually we tried, first of all, to study to develop the mathematical model to study the vaccine-induced adaptive immune response. So through the cell-mediated and antibody-mediated actually immunity. And then we investigated the effect of different prime boost strategies, including booster delay and sparing by quantifying their effect on the immunological outcomes. So actually we investigated these strategies effect by delaying the second dose and also by reducing the second dose amount on the immune response, and see that whether we will have the enough immune response to fight with SARS-CoV-2 virus or not. So I'm going to start with the schematic figure here that you can see when the vaccine particle are stimulating the adaptive immunity. So actually the vaccine particle here, you can see can stimulate the adaptive immunity through 3 different signaling pathways. So the first one is that when the antigen presenting cells actually are presenting the pathogen through MHC1 protein to the precursor satellite cells. And then they are activating the the precursor sorry, I forgot the cytotoxic T cells. I'm sorry. So these are the precursor cytotoxic T cells, and then the cytotoxic T cells that are the killer cells that are killing the infected cells after activating. So then, so they first, through this signaling pathway, they are activating and started killing the infected cells. The second pathway is that when the antigen presenting cells, it is APC2 because they are presenting the pathogen through the MHC2 molecule to the T helper cells. And then through the activating of the T helper cells, so they are actually, the T helper cells are differentiating to Th1 and Th2, that the differentiated forms of the naive T helper cells. And then, yes, they are killing the virus through, actually they are first differentiated to Th1, and then the Th1 are secreting some cytokines and then activating cytotoxic T cells. And then through cytotoxic T cells, they are killing the infected cells. And at the same time, they are activating Th2, and Th2 are secreting some cytokines, and then they are activating the B cells. So then the B cells are differentiating to the plasma cells and the memory cells, and the plasma cells are generating antibody, and then the antibodies are killing the viruses. So this is the main signaling pathway after vaccine injection. And also the third one, when the naive B cells are encountering the antigens, and again, they differentiated to the activated B cells. However, through our paper or our work, so actually we looked at the main actually signaling pathway. So we assume that, and we actually in this model, we try to simplify it as much as we can, and we ignored some details in some other details, and just we looked at the main compartments. So we assume that the vaccine particle actually are activating the T helper cells, and the T helper cells are generating the interferon gamma. Interferon gamma is the main cytokine in the adaptive immune response. So the interferon gamma is activating the cytotoxic T lymphocytes. And also they are encountering with the vaccine particles directly. And then they are activating and killing infected cells. And also on the other hand, you can see that the T helper cells are secreting the interleukin-6. Again, is the main cytokine, is the main interleukin to activate the plasma B cells. We assume then we actually ignore the precursor B cells or anything, and then we assume that they are only directly activating the plasma B cells, and then they are generating the antibody. So through 7, I think, simple ODE equations and using the heat functions and the mass action term, so we developed a mathematical model to simulate the adaptive immunity. Then after that, we had some parameters. Some were chosen, some were coming from the literature, and some others were fitted when we fit actually our model with the AstraZeneca data from the Oxford data. So then we can see here that our model was compared with the experimental, with the clinical data. So the blue curve is for the first dose injection. So, and the second dose is the red curve. So we can see that there is a good agreement with our data here and also our model prediction. And so we had 2 different sets of the data, one for the SARS-CoV-2 injection, and the other one is for the interferon gamma response. So the blue and the red curve, again, is for our model prediction, and we compared with experimental data here. So again, so after fitting here, we can see that the second dose also has a good agreement with data. So we predicted our parameters, and then, yeah. So in the next step, our aim was that to investigate actually the different schedules and then actually looking at the immunoresponse for different schedules for the different region, different actually strategies. So the first one is the SDSD schedules. So it means that the first dose and the second dose has the standard amount of the vaccine particle, 1,000 vaccine particle we assume in the first and second dose. However, we looked at the antibody response for the different prime boost time interval from week 4 up to week 20. So what we can see is that even enlarging this time interval up to week 20, we can have the higher secondary antibody response in comparison with the primary response. So this one is the primary, and then after the second dose injection, the antibody response is higher than the primary response for the SDSD actually regime. Now, if we compare, if we reduce the second dose as the limited dose, so actually we decrease the second dose by half, so we have 500 vaccine particle here. So when we decrease the second dose amount for the 500 vaccine particle, we can see that after week 8, actually here, after the green one, so the secondary antibody response is less than primary antibody response. And even if we compare with the third schedule, when the second dose is reduced by quarter, we have in the second dose, we have 250 vaccine particle. We can see after even week 6, so the secondary antibody response is lower than the primary antibody response. So our first results from the model prediction is that, actually the immune response corresponds to the vaccine particle. The higher vaccine particle, the more stimulated antibody response. We also looked at the cytotoxic T lymphocyte response. So we can see that again, we looked at the through different strategies. Delaying different delay times in prime boost. And so the first regime and the second regime and the third regime here. And we can see that if we look at the compare the highest amount of the satellite cells that are, cytotoxic T cells that are activated, we can see that for the first one is about more than 100 cells per milliliter are stimulated in comparison of the two other regimes. So again, we can see that even the cytotoxic T lymphocytes are more activated when they are receiving more vaccine particles. This is also another evidence that the immune response is actually attenuated with the less vaccine particle that they are receiving, and they correspond with the vaccine particle. Now, the main question was that whether with these schedules that we are trying to overcome the shortage of the vaccine, because delaying the prime boost and reducing the second dose amount can help to have enough vaccine, enough vaccines for all peoples, and also gives the time for the vaccine companies in order to generate the vaccines. So yeah, in order to answer that is that whether we have the enough antibody or cytotoxic T lymphocytes. So actually our model cannot answer this, but what we did is that we compared our model prediction with the clinical data. So from the clinical results of 175 recovered COVID-19 patients, we can see that 39 patients have high medium levels of antibody, which is between 1,000 and 25,000 titers. So if from our model prediction for the SDSD actual regime, the minimum promoted antibody from our results is more than 5,000. which is in this interval, which is between 1,000 and 25,000. And even for the SD-LD regime, with the LD, which is decreased by half, the minimum promoted antibody is more than 3,000. And for the third regime, it is more than 1,000. So then our model predicted that decreasing the dose amount and for the by quarter, we will have enough antibody to fight with the SARS-CoV-2. So in summary, we can say that either limiting the second dose or increasing the prime boost time interval leads to attenuated adaptive immune response, as I mentioned. So it is corresponding to the vaccine particle. And stretching the prime boost interval to 18 or 20 in ST-ST or ST-LD regimes, decreasing the dose amount by half or by quarter, the minimum promoted antibody response is comparable with the neutralizing antibody in the recovered COVID-19 patients. So then this was the main results of our paper there. So, and I also would like to talk about a little bit about our current projects that currently I am working on finalizing two projects. The first one is the immunoepidemiology project that we are looking at how the immunodiversity can affect on the disease transmission. And then the second one here is we are actually developed a mathematical model to study the B lymphocytes and the SARS-CoV-2 neutralization antibody. So in this schematic figure here, you can see that it's very specialized on the B lymphocytes. So in response to the SARS-CoV-2, actually, the naive B cells are activated, and then they start the proliferation and the differentiation. So the germinal center B cells. So the germinal B cells in the germinal centers, in the light zones of the germinal centers, are getting some cytokines from T follicular helper cells that are interleukin-4 is the main one, and interleukin-21. But I have ignored interleukin-21 because we couldn't find enough data for that. And also at the same time, they are encountering the antigen through B cell receptors. And then the germinal center B cells are differentiating to long-lived plasma cells, memory B cells, and start the proliferation. And also we looked at the post-secondary response, the post-secondary infection response. So in the secondary infection, the immune system is activated, and then the memory B cells again start to differentiate to the long-lived plasma cells and the germinal center B cells. And then again, the germinal B cells are coming back to the germinal centers and again to the circle. So, and then, yeah, we developed our mathematical model to simulate these B lymphocytes and then actually antibody-mediated immune response. And then, and we investigated some parameter ranges because for the proliferation, actually for generation of the antibody by the plasma B cells. So because we had to do some assumptions and then, but however, we couldn't find those parameters. And then, so actually our model predicted some ranges for some main parameters actually that we found from the global sensitivity analysis. So yeah, then I would like to thank my supervisor, Professor Morgan Craig from University of Montreal, and also my Manitoba supervisors, Professor Portet and Leo and Arino, and my collaborators from Ryerson, Dr. Volke and Dr. Heffernan and Moines, and our lab members here. Thank you.
Okay, thank you, Suzan, for the talk. That was a great talk. Thank you so much. Yeah, so we want to give people the opportunity at this time to ask questions and also discuss what their views are on this project. So yeah, I can see one question here in the chat, so I'll go ahead and read that. But after this, if anybody wants to unmute him or herself and ask the question directly, we encourage that. But if you also want me to read, you can put it in the chat and I can read that for you. Okay, so the question is, may I ask how the model captures the non-linearity in data in the first part of the talk?
How the model captures the non-linearity in data? You mean for the chemotherapy?
Yes, the first, please. You said that you have both linearity and non-linearity behavior in the data. So yes, I can see, as far as I understood, you mentioned that the model can capture both cases, linearity and non-linearity. I was wondering, I know that you didn't talk about the mathematical parts, but I was interested in that part.
So actually, both of them are non-linear. I don't know what you mean with the linearity. So here, even there is an oscillation here, it is because of the 5 days on and 2 days off holidays. So we can see some oscillations here, right?
Yeah.
So both of them have the non-linear behavior, but here it is some oscillation. Yes, our data captures that. And then here also our data captures this behavior, this reduction. So, yes.
I mean, I was talking about your model. How do you build your model to capture this behavior, this kind of nonlinear behavior in data?
So yeah, after developing our model, we use the optimize function. First, we fit our model. I think our code's in Julia, and then we use the optimize function in Julia. Yes, and then fit my model with experimental data. First with the daily, and then with the 5 days on and 2 days off. And then, so yeah, it is the results of the curve fitting.
Okay, thank you very much.
Yeah, I don't know if I can understand. I know that maybe it's, you mean that it is maybe, you know, but we can see that after using the curve fitting, doing this.
Yeah, maybe it's better that I read it, because I wanted to see the mathematical model, but but it's better that I read the paper. Thank you very much.
You're welcome. But the mathematical model, yes, you can look at in the paper, maybe just, yeah, because I didn't include that. Yes, the model here.
Thank you.
Yeah, should be here in this paper.
Sure, people still have the chance to ask their questions.
Seems that no question.
Yeah, it looks like everyone is cool with the talk. Yeah, that means you've done a great job. Thank you, Dr. Suzan Farhang-Sardroodi. We probably want to end the session for today, and we want to thank everybody for tuning in today to join us for this great seminar. Looking forward to seeing you again in our next seminar. Thank you, everybody. And thank you, Suzan, for your time and the talk. Really appreciate it.
Thank you, Joseph. Bye.
Bye.